So Vinita's problem we still have managed to sort out.
She still not managed to sort out. So Pinaki says we can start at 3:30.
Yeah, this prompt is coming that connecting to audio. Sometimes this connect with computer audio, that window goes underneath the Zoom window. So if that is hidden, one has to see that might be a problem.
I'll just message.
If there are more than one, I mean, if there is a microphone, then that is a different issue. But if computer audio is the one, then it will work perfectly.
Vinita says it shows earphones instead of the usual icon. That's strange.
If she can share the screen, we can go to the zoom.us, the menu, and go to the preferences. And if you open the preferences, in the audio section, the test speaker, right after test speaker, there are options of choosing the speakers. So if she's using a microphone, she can choose that instead of the computer audio. That might help.
Quite follow.
Yeah, that's a good suggestion. Seems to be working here.
Do you want to send that as a direct message to her? Because
I can, let me have a number I can try and call.
Yeah.
Vinita, on the Mac, if you can go up to the Zoom thing right at the top.
Sir,
Zoom khula hua hai na?
Yeah.
There's some right on the top layer. There's something called zoom.us, meeting view, edit view, etc., etc.
Okay. Gautam, can you tell Vinita that there's that headphone icon that she said is there? If she clicks that, then there will be a join audio option.
You're saying, one sec. Sorry, Rahul. What were you saying again?
If she clicks the headphone icon, then there will be a join audio option, and then she can click that.
He says if you click the headphone icon, there will be a join audio option, which you can click. Are you able to see that? She's tried that, but she says it makes no difference. So she'll just try a fresh Zoom app and see what happens.
Okay, should we be starting then?
I can give you I can send her number to you.
Yes, I think let me introduce you and you start off, and I will try and sort it out offline then. We'll just check whether Gayatri can. We'll start in half a minute.
Hi, can you hear me?
Hi, I can hear everybody now.
Oh, perfect.
Thank you. You want me to share, right? Sorry, that was the idea.
Just to test.
I know. Now I have to do everything again because everything was closed down. Wait.
We just assume that it works, Vinita. Just set it up because let's start now since we're a little
Well,
late.
you can start. Sure. No problem.
You may have to stop sharing.
Yeah, I will.
Rahul,
Okay.
you want to start?
Yes. Okay. Let's start now. So, good afternoon, everybody, and welcome from my co-organizer Pinaki Chaudhary and me to this mini symposium on looking back, looking forward. I should credit Gautam for that title. So we are running a little late, so I'm not going to waste your time, but I'd like to quickly thank our director, Professor Ravindran, who just took over a couple of weeks ago formally for egging us to do this repeatedly. I'd like to thank our media team, Gayatri, and our outreach coordinator, Varuni, for the help that they have done in bringing this together. And I'd like to thank all the speakers for agreeing promptly to speak and to Gautam for agreeing even more promptly maybe to moderate it. And with that, I'll hand over to Gautam. Thanks, Gautam.
So welcome to all of you to looking back, looking forward. This is, in a sense, the true aim of ISRC to sort of provide a forum for scientists also to interact with each other and to see how the narrative regarding COVID-19 has now changed. We now know a lot more than we knew last year. And we also, in some senses, know a lot less than we knew last year. We know what we have to be confused about now, whereas earlier we weren't even sure what we ought to be confused about. So so we have 6 wonderful speakers today. We have Uma Ramakrishnan, Vinita Bal, Dheeraj Hazra, Rajesh Sundaresan, Chitra Pattabhiraman, and Vinod Scaria. All of them outstanding in their own field. And this will give you a scientific coverage over a bunch of areas, all related to COVID-19 in India. So I'm going to start in that sequence of speakers that I listed. I'm going to introduce all of them briefly, let them speak for about 10 to 15 minutes. Then we move on to the next speaker. And once we're done in about an hour and 10 minutes, then we'll have a question and answer session. At the end, you can address your questions. The audience can address questions through the chat box or the Q&A. And they will be screened and then passed on to me so that I can address them to the individual speakers. So let me start with Uma. Uma is at NCBS Bangalore. She's an Indian molecular ecologist. She is dean currently at NCBS, I think very recently. Her research investigates population genetics and evolutionary history of mammals in the Indian subcontinent, in particular tigers, which she's famous for. She's been interested in looking at One Health approaches that integrate ecology with human health, and that's the context of what she'll talk about today on biodiversity, humans, and disease spillover. Go ahead, Uma.
Thanks, Gautam, for the introduction, and thanks the organizers for inviting me here today. I'm very happy to be here, and I'm going to start by sharing my screen for my presentation. Okay. So as Gautam mentioned, I'm at NCBS, and he also mentioned that I'm a molecular ecologist, and I've spent a large proportion of my life working on endangered species. And so you may wonder, why on earth am I here as part of a symposium or mini symposium speaking about COVID? So in my exploration of, trying to understand human impacts on biodiversity, I realized that a very strong interface for potentially negative consequence for both biodiversity and humans is the human-wildlife interface, especially in the context of emerging zoonoses. And this is something I have been trying to investigate further over the last 5 or so years. And so that's what I'd like to share, some of which with you today. So, of course, this is something you can probably recite in your sleep, but just to remind us all that 75% of all emerging infectious diseases are zoonoses. They spill over from animals, mostly from mammals, sometimes from birds. Why mammals and birds? Simply because they're physiologically closest to us, evolutionarily closest to us. And so pathogens or microbes which survive in them can then quickly jump over to us and similarly reproduce. And these emerging zoonotic diseases can spill over from various, in various ways. Live animal markets have been discussed a lot in the last year. Of course, intensive wildlife farming, ostrich farms, civet farms, and so on. We heard a lot about the not the case in Denmark where in the Netherlands where many animals had to be sacrificed because of being infected. And of course, domestic animals, for example, pigs, cows, horses, Hendra virus, for example, spilled over from horses via bats to humans, and wildlife hunting. So I basically am interested in this interface between wildlife and humans, the direct interface between wildlife and humans. Why is this relevant for us in India? Well, there are several meta-analyses which investigate probability of spillover, across the world. And these analyses suggest that there are several correlates of spillover, and these are high mammal biodiversity, high land use change, or basically change in the parcels of land, whether it's agriculture or forest, change of forest to agriculture, change of agriculture to built up areas, and so on. That's land use change, high human population density, and high livestock density. So India scores very highly on all of these. We have high mammal diversity, high land use change, high human population density, and high livestock density. So you can see then that across the world, India and China basically light up as areas of high probability of a zoonotic spillover. So this makes it important for us as well to understand these human wildlife interfaces and understand probabilities of spillover in the Indian subcontinent. So where is biodiversity high? Let's break this down a bit. So of course, you can see this is a map of vertebrate diversity. It's a index called species richness, which is simply the sum total of species, the number of species in a particular location. It's a beautiful map. I love maps. But this map actually shows you that we see high species diversity or richness in the tropics. We also tend to see high diversity in geological features like mountain ranges. You can see the Himalayas and the Western Ghats are lit up. But also to remind you that actually speaking, species richness or the number of mammal species is much higher in Southeast Asia as compared to India. So comparing this map to the earlier one, right? Southeast Asia was not so high, so lit up in terms of risk of spillover. And that's because while India does have fewer mammals than does Southeast Asia, the land use change in India and the human population densities in India far surpass those in Southeast Asia. And so as a result, overall, India becomes a much more high risk area for spillover. Okay, so if you look at India itself, I mean, that map is a little bit difficult to see because it's global. How does species richness, for example, of known reservoirs, we've heard a lot recently about bats. How does the diversity of bats look across India? Are there equal numbers of bat species across India? Do some parts of India have more species? And you can see immediately that northeastern India has much higher diversity of bats than do other parts of India. And again, the Western Ghats to some extent has high diversity. We know, for example, that India has some biodiversity hotspots, 3, and if you include the Andaman Nicobar Islands, 4, and the Northeast is one of them, as is the Western Ghats. So basically, as we would have expected, areas which are biodiversity hotspots tend to have more bats, tend to have more rodents. Also, small mammals like rats and mice are known to be reservoirs for several viruses, but also many, many bacteria, bacterial spillover agents. And so you can see that immediately, if I was to prioritize an understanding of spillover risk, um, which you can hope to use such knowledge for mitigation, one would tend to study these questions or problems in biodiversity hotspots, which are the Northeast and in the Western Ghats. Okay. So before we just move on a little bit to some of the work that I have done, I just want to also us to look at where have we seen outbreaks in India. Okay, so this is a map we put together, and this is just for 1 year, um, from the IDSP, which is the Integrated Disease Surveillance Program for India, and the IDSP maintains records of outbreaks across the country. Unfortunately, it's like a PDF, but you can actually download all these records, and that's what we did. And then we mapped kind of outbreak occurrence across India. And you can see, for example, that some outbreaks tend to be very broadly distributed geographically, like dengue or chikungunya, whereas others seem to be more restricted. Some tend to be forest associated, like scrub typhus or rickettsia. But surprisingly, maybe this surprises you, it seems like the Northeast doesn't seem to have that many kind of labeled outbreaks. They don't seem to be a lot, right? Definitely the Western Ghats does seem to have a relatively high number of not just circles, but different colors of circles, indicating a diverse set of outbreaks, which are, kind of co-occurring there, which is what you would predict based on the high biodiversity and also the high risk of outbreak because of human population density and land use change, right? So that's interesting. Um, I don't know the answer, but is this because of underreporting in the Northeast? It could be. It could also be that there are unknown pathogens in the Northeast, spillover events or small outbreaks which go undetected and maybe unobserved. But, that's what I would like to think. But, we'll have to see, we'll have to interrogate this with data in general. Okay. So what we have been doing is, I hope I've convinced you so far that studying the human-wildlife interface in the context of India to understand emerging zoonoses is critically important, right? Globally, from a global perspective, as we've seen from the last year, it's not important just for us, but for the world. And also that there may be some places which might be better sites to do such studies than other places, right? So when you start something so exploratory like this, you don't want to just, I don't want to just go outside my, you know, whatever apartment and start catching bats. Maybe that doesn't make sense. So what we did is basically we've done basically two types of studies. One is in the Eastern Himalaya, basically studying a bat harvest in Nagaland, and I'll show you a bit more about this, as well as in the Western Ghats, where we're looking at fragmentation. You've all drunk tea from the Nilgiris or had coffee from the Western Ghats, Robusta or whatever coffee. And so you know that these landscapes are extremely fragmented. And I'll show you what this looks like in a little bit. So this is a picture from the village Mimi, where we've been working for the last 8 or so years. You see in the background the Bomar, who are a community of Nagas, and they have an annual bat harvest where they smoke a cave, and as the bats are asphyxiated, they're coming out of the cave, the bats are killed. And you can see in the foreground here on the ground those are all dead bats. So on a given day or 2 days, the bomber in this traditional harvest kill between hundreds to thousands of bats. And this has been going on for many hundreds of years, at least according to their cultural storytelling. To the best of our knowledge, there has been no major outbreak here. People are healthy, nobody is sick or any such thing, neither are the bats sick. Yeah, but at the same time, this is a high risk interface, because as the bats are coming out, they are getting cut, they have cuts, and the people have cuts because the bats are biting them. So there's an exchange of fluids possible, and also very close contact with high densities of bats. So this is, we thought, an interesting place to look for, what are the RNA viruses bats carry, and is there any serological evidence for spillover in both the bats and the people. And actually, before we started this work, we collaborated with Dr. Rita Mani from NIMHANS, who looked for serological evidence of Lisa virus in bat brain tissue. Lisa virus is the family which includes rabies, and basically she at that time, in 2017, working with us, did find serological evidence for some exposure to Lisa virus. So I'm not going to show you all the results because this is a mini symposium, but what we have found so far, and this project is still ongoing, we've had some hiccups and had to stop for a while, but basically what I think it's a really interesting set of samples and context, and what we've found so far is that there are potential pathogens circulating in the bats which are consumed by people in northeastern India. We found serological evidence for 3 unique filovirus families in the bats and 2 families in humans, which was basically shared. Like, of the 3, 2 of the viral families showed shared serological response in bats and humans, right? When you say humans, we sampled the hunters and other members of the clan who are involved in the harvest. Okay, and this suggests then spillover because it is shared serological response. Both the bats and the people show some response to something, but it's the same thing, right? So it seems like this is a good system to study spillover. That was what we were initially just exploring, and as of now, we are screening for other viral families. We didn't find any genetic material of any filovirus, which is good. But we are looking at many other viral families like astrovirus, paramyxovirus, coronavirus, and so on, and basically trying to see whether we have genetic material from these. Now, from various studies recently that coronaviruses are extremely diverse, right? And we'll get back to this towards the end. But bat coronaviruses are very poorly explored. And so it's quite likely that there will be a lot more diversity to explore. This need not be pathogenic diversity. It is just microbial diversity of viruses and bacteria. Whether they are pathogenic or not is a different question, right? So just quickly about our work in the Western Ghats. This is a shot from where we're working. It's a plantation called Kadumane. You can see here the sholas, or the higher altitude grasslands, and the very special high altitude forests, which represent the Western Ghats. The Western Ghats has been subject to a lot of defaunation, which means naturally imagine the Western Ghats before people colonized it, and mainly before the British started tea plantations. There was a lot of habitat, and all of this habitat was filled with large carnivores, but also large ungulates like gaur and sambar and deer, cheetal, and so on. But defaunation is the loss of this large wildlife, the large ungulates, and the loss of these ungulates changes the community. It results in what is called rodentication, that is an increase in density of rodents, rats and mice. When you lose things like bison, you have an increase in the density of rodents. So what we are looking at in the Western Ghats is basically, this is just a picture of field work. We catch rats both on trees and on the ground with these traps called Sherman traps. We can process them. We don't kill them. We just look at, various, we sample, blood with retroorbital, the capillary, and then, we just let them go. So we can actually then look at, again, in this case, not serology, but we can look for occurrence of specific bacterial agents. And in this case, we explored a bacteria called Bartonella. But basically, just to show you that picture looks very pretty, but overall, this is what is fragmentation. This habitat is in small little bits of parcels of, grassland, forest, tea, abandoned cardamom, and so on, all kinds of stuff. And you can imagine that the rodents which live in these different habitats differ. Grasslands are dominated by mice, and forests are dominated by rats. And so you can imagine that this kind of interface can lead to a lot of spillover even between the different rodents. Okay. Um, so basically what we found is overall a very high prevalence of Bartonella. Between 50 to 80% of rodents uh, yielded genetic material for Bartonella. And Bartonella is not a very bad pathogen. It causes, some of the lineages are pathogenic, cause scratch fevers and so on. Something you may not even know that you have Bartonellosis, right? It's very mild, probably. But overall, there are people in these plantations, tea pickers in the labor lines and so on. So it again suggests a high interface for potential spillover. Of the different lineages of Bartonella we didn't sequence whole genomes, but maybe a third of the Bartonella genome. We found that there were 5 pathogenic lineages that were in, so our like the Bartonella associated with our rodents nestled within known pathogenic lineages, some of them. And we also looked at vectors, and we found that some of these vectors, and these are basically mites, which are arthropods in this, like in the spider family. They're very difficult to identify, but we just, you just crush them and you can extract DNA and get Bartonella DNA from them. Were also potentially the vectors for this particular bug. So all that is fine. Like, I'm just giving you some stories, but what do we do with this information? Um, I guess, of course, there is some scientific interest, but I think that understanding the distribution of these different lineages and occurrence of these potential pathogens can help us to really understand not just hazard, like how many bats have how many coronaviruses, but also exposure and vulnerability. And at the interface of all that is risk. So for example, we know with pathogens like Nipah that bats shed Nipah for a very short period of time, right? So if we can narrow down when that period of time is, then we can minimize exposure of people at that time, right? And that will minimize the risk. So obviously it's not a solution to kill all the bats or destroy all the caves, but we need to learn to live with the pathogens among the biodiversity that subsists around us. And unless we understand its distribution and change over time, that will not be possible.
Vinita? Vinita, we can't hear you.
Now?
Yeah, now we can.
Okay.
Yeah, go ahead. You better go back to the first slide.
Yes. This is not going to work. The slide share doesn't work. I'll have to go back to sorry, this is the usual problem, but I'll not do the.
Yeah, that's fine.
Yeah.
Yep.
All right. So going back still, this is the first slide. So how do vaccines protect from infection and disease is what I'm going to talk about, and of course in the context of variants. And since this is a mixed crowd, I'm first going to make a distinction between infection versus disease, because that is actually relevant. Infection is when a pathogen, an organism invades our natural barriers, and infection invariably will trigger off in immune response, what we call both natural or innate immune response as well as adaptive immune response, which means involving T and B cells. But this doesn't necessarily mean that when infection occurs, we feel sick. When we feel sick, or that's the condition that we call as disease. I know this disease is spelled with I, but it is dis-ease. When you are not comfortable, you are complaining because of infection. That's the infectious disease state that we talk about. And many more infections come our way, and most of them go without development of disease. And the immune response, which is generated against the infectious agent, is the one which normally prevents from the development of disease. But also immune response is directed against multiple components of protein, carbohydrates, and so on and so forth of the infecting agent. And only a small component of the whole universe of immune response against a pathogen constitutes what we call as protective immune response. And this protective immune response is different for different kinds of pathogens or different kinds of organisms which come our way. What you see at the top is free living, meaning outside the cells or pathogens. And these pathogens are normally tagged by antibodies and complements. And once they are tagged, then phagocytic cells, which are the cells which eat up every particle that they come near. These phagocytic cells, mainly neutrophils and macrophages, clear the pathogen, as has been shown here. So they are engulfed and killed off. So that's the contribution of antibodies and complement. The second line shows helper or CD4 T cells. And these CD4 T cells tend to activate these macrophages and neutrophils so that intracellular pathogens, intravacuolar pathogens, I should say, they are killed off. But you have to remember that these pathogens are inside a vacuole, and hence just getting rid of the vacuole tends to spare the cell but kill the pathogen. The third kind is the cytotoxic or CD8 T cells. And here, the whole cell is involved because the pathogen is not sitting in a vacuole but sitting inside the cytoplasm. As a result, the best option available is to kill the organism. So these are the three broad kinds of immune responses. All three contribute to protection in different situations. And this is the continuation of the earlier slide. And the nature of antiviral protective immune response, which can be triggered, of course, by infection and or should be triggered by the vaccines. And it consists of neutralizing antibodies as the first point. And this neutralizing antibodies normally bind to a very critical portion of the pathogen so that entry of the pathogen or pathogenic virus inside the cells is prevented, thereby downstream consequences. This doesn't always happen in 100% of the cases or 100% efficaciously, so that viruses can get inside the cell and they can produce infection. killing of such virus-infected cells, or which are cytotoxic or CD8 T cells, as I showed you in the previous slide, this also forms a protective immune response. The third component here mentioned is the helper T cells. These T cells tend to improve the quality and quantity of the antibodies produced, such as the neutralizing antibodies. So all three components are important, but to lesser or more extent. This is something that I'm going to explain in the context of variants. And this is a textbook picture, but textbooks also tend to show you a lot at times. And what this shows is this center portion is a portion of a protein or an antigen. An antigen means anything that can trigger an immune response against it. So this antigen has small units called epitopes, which are shown at that four corners. These are called epitopes, so they are smaller parts of the antigen. And against each epitope, a complementary antibody is formed. And these, obviously, this is the antigen binding site of the antibody. And this shape is different from what you see for the other antibodies. This is a crude picture, but essentially any antigen can actually trigger many antibody responses, as has been shown in this. And the receptor binding domain of the spike protein of this SARS-CoV-2 virus is again an entity which will have multiple epitopes, and it will trigger many neutralizing antibodies. And when I say many, it's not just quantity, but it's a polyclonal response. So each one is a clone. There will be many replicates of this clone or this antibody, but there are, as it is, polyclonal, multi-clones response which is there. So this is something that we need to remember before we talk about the variants. So let me mention about the viral variants, and I'm not going to necessarily go into the details, but the variant which caused major epidemic in the last 2-3 months is identified as B1617, or nowadays it is called Delta. So this is not, I mean, there are Delta and its further variants also of the 617, but let's just assume it is Delta that we are talking about. As compared to the original Wuhan virus, or as compared to the B117, which was the UK variant, B617 seems to have a much better transmission capability, which means the ACE2 receptor, ACE2 receptor, which is expressed on many cells, including epithelial cells, the outer lining of the nasal epithelium, for example, nasal lining. These ACE2 receptors are binding this Delta variant with a higher affinity. So obviously, the receptor binding domain of Delta is somewhat different from receptor binding domain of the original Wuhan virus. And many of you, I'm sure, are aware of this. This I simply wanted to state as a point. So increased affinity, as I said, with that increased affinity, there is increased binding, increased entry, and hence for the much more efficient replication that can take place, so that people infected with B1617 can actually have possibly higher load. They can spread it to each other as well as within the cells inside the body. So mutations, when they take place, can change an epitope. And this is where the previous slide that I had shown you of multiple epitopes comes into picture. So mutations do not change the whole protein, such as receptor binding domain. So what we are seeing with the Delta variant is one or maybe two changes of the epitopic kind within an RBD. And I showed you only a two-dimensional picture of to explain the epitopes. You can imagine that there will be many more epitopes on the receptor binding domain. So when neutralizing antibodies are generated, these neutralizing antibodies of a variety of kind, all of them are binding to RBD, and it's a possibility that 617 variant has changed the structure of RBD in such a fashion that out of, say, hypothetically 15 neutralizing antibodies as polyclonal response, maybe one or maybe two have been changed enough so that the antibodies against those epitopes are not capable of binding to 617 any longer, and hence the total neutralizing capability of the of the serum of somebody who's vaccinated or who was infected and has recovered will change. So the point that I'm saying is that neutralizing antibody potential is not lost completely with a minor change in the viral genome. And however, cumulative changes over a period of time, as we are going to see, will result in cumulative loss of neutralizing potential. This, in addition to that, I had mentioned that there are also CD8 and CD4 T cells which contribute to immune response, but for the sake of time, I'm going to skip this slide, though again, the same logic that I mentioned for antibodies also operates for this. So now let's come to the last point that I want to make, or a series of points. What is the utility of SARS-CoV-2 vaccines against the original as well as the variants? So what we know for sure is that for even a single dose of any vaccine will provide more protection than unvaccinated, uninfected, and hence non-immune persons would have. So we have been talking, and there has been a raging debate whether we should try to cover every individual eligible for vaccination with at least one dose, or should we be covering every individual with 2 doses? So if it is 2 doses, the single individual who has 2 doses will certainly be protected. However, as a public health measure, it is always better to have many more individuals protected, and we have to consider this. We have to consider this seriously, because in India, as we know very well, there is a massive shortage of vaccines. So covering as many individuals as possible can be one target which we may have to look forward to as a compromise. If we had enough doses of vaccine, enough manpower to vaccinate very, very rapidly, of course, 2 doses to every individual would have been the ideal situation. So the second point says that is about the relative risk from the vaccine versus the infection. There has been vaccine hesitancy, and that is even more common amongst the middle classes and upper middle classes for one reason, and for another reason, like inability to access COVID application for registration in the lower socioeconomic strata who do not have smartphones, for example. However, what we know is that the death rate following vaccination is at least 3 logs lower than that following infection. So current case fatality rate is closer to 1.5 to in different areas, but total for India is just short of 2. We are talking about case meaning every person who is admitted to the hospital or at least has been RT-PCR. There have been many asymptomatic infections, and that is the infection fatality rate, which is even smaller than the case fatality rate. So 3 logs lower is for the case fatality rate. And this protection, of course, that is generated after the vaccination can and will be extended to some variants in the near future, based on the logic that I said, that all the different neutralizing antibodies generated will not become defunct with 1 mutation or 2 mutations. Eventually, when there are cumulative mutations, like what we are talking about Delta Plus now, which is an earlier mutation which got added more or less to the Delta mutant, such cumulative mutations might actually result into further decrease in the vaccination mediated protection, and we may need another booster with another construct of mRNA or another construct of the adenovirus mediated spike protein. And what is likely to happen, so that's the last point that I want to make, is that virus eventually, including SARS-CoV-2, may attain the optimally infective state after many mutations. And this might be the steady state, in the sense any further mutation will actually decrease its ability to infect, to spread, and remain quote unquote alive in the population. So this is something that we need to understand. And that is why, while everybody is really in a panic situation, I shouldn't say everybody, I'm sorry about it, but many people are very worried about the Delta plus wave. But currently, we are not at a stage when we can say that the Delta plus virus is going to produce even more havoc in the population because of whatever mutations that it is going to say. I thank you very much. I hope I haven't exceeded my time too much. I'll stop sharing this.
Thanks, Anita. Okay, so now let's move on to the next speaker. And our next speaker is Dheeraj. This is Dheeraj Hazra, who works at IMSC Chennai. He's been a faculty member at IMSC for about the past year, I guess. He spent many years in Korea, Italy, and France, following a PhD from Harishchandra Research Institute in Allahabad. He works in essentially numerical methods in cosmology, but his special skill is the sort of numerical diversity that he brings that really powered the whole InSystem model and the numerical approaches to that. Dheeraj, go ahead.
Thanks, Gautam, for the generous introduction. I'd like to thank the organizers. Is my screen
Yes, it is.
visible
It is.
full screen, the full screen mode, right? Okay, thank you. So I'll be talking about the InSystem model for COVID-19 in India, regarding mainly about the preprint that we released in the beginning of June. The InSystem team is composed of these researchers from different institutes, from IMSC Chennai, Pune University, and IISc Bengaluru. The whole story started with Professor Gautam Menon and Professor Pinaki Chaudhary initiating this InSystem collaboration in the beginning of, I think, the middle of March 2020. So since then, we have been working on building the models and analyzing the data. And as a result, in 2020, we had released a series of analysis. For example, in the first version, we released the model, we discussed the model and several lockdown strategies. Then after that, we mainly concentrated on periodic lockdowns. And after that, we wanted to have certain estimation of the national lockdown for the 68 days that we had. Then we tried some projections. For example, we did some Chennai projection and Delhi projection, which turned out to be quite successful. And later on, we moved our interest, we shifted our interest to the data analysis, and the works that we had done, the analysis that we had made, resulted in this preprint, as I said, was released in the beginning of this year. And let me just take a laser pointer. Okay. And at the same time, we have been doing projections for different states, different districts, and we have been working with different technical advisory committees for different states. For example, we did projection. We are doing regular projection for Andhra Pradesh, and Farina leads that projection. We have been doing projection for Karnataka, the districts of Karnataka and the Karnataka state as a whole. And Pune projections, we also did Maharashtra projection, which Balchandra leads, and we have been also doing the projection for the national data. All right. Now, the first question, when you say that we are trying to model the COVID-19, the first question that comes to my mind is that why we would like to model. There are data available, right? For example, here I have taken the screenshot from COVID-19india.org, where I have just shown daily confirmed cases, active cases, recovered, deceased, and tested cases. So if we have data, what is the role of the model? Firstly, this data shows what we have tested, right? So we cannot say that the people from whom we have not tested, if we have missed some infection. So if there are bias in the detection, all right? If we are undercounting deaths, so first one was undercounting infection, then we can also model and find if there are death undercounting. So these are some things that the data cannot tell you. Secondly, the data cannot extrapolate beyond the ranges, right? So you need a model to do some realistic projections, and often model can do that. We can also help in making policies. We can provide suggestion for policymakers. For example, we can trace that if the lockdown for certain duration, if extended, if the testing is extended, or for example, the vaccination strategies, if we vary our vaccination strategy, what happens if whether we are basically reducing the infection, reducing and the we are stopping the progress of the pandemic, or we are basically making the deaths smaller. So these are some questions that we can ask from the model. And finally, from one wave, let's say, we can establish what was the underlying dynamics that for the progression of this pandemic. All right. So we have established that we need a model, not just the data. Now the question is that whether what properties should a good model contain? So this is the basic epidemiological model that everybody knows. For example, this is the SIR model, susceptible, infected, and removed model. So here, the question is that whether this model can address the COVID-19 pandemic. The answer, simple answer is no, because from the beginning of the pandemic, we knew that this pandemic had few properties. For example, we had asymptomatic infection, and the people who are symptomatic, they can be subdivided into two categories, like mild and severe, the people who need medical care and who doesn't. So, and another thing is that we should reproduce, the model should reproduce the observables. Let's say I want to compare our model with the data. Now we need to have those compartments. For example, the infected should be there. We should have a compartment for the hospitalized category, and we should have a compartment for the diseased. So that's how we build the model. So SIR, for example, now can be divided into SEIRs, susceptible, exposed, and then the rest. Then the SEIR can be further divided, the exposed category. So exposed are basically the people who are exposed to the infection, but not infectious. So now after that, they are subdivided into two categories. One is asymptomatic, one is pre-symptomatic. Later on, the pre-symptomatic can be subdivided into mild symptomatic and the severe symptomatic. They all go to the removed compartment. Now, I can also say that, okay, this severe compartment goes to the hospitalized compartment. And now, since we are now focusing on to compare with the data, we can subdivide this R, or the removed compartment, between two parts. One is recovered and one is dead. All right, from hospitalized, people can either recover or can die. So this is the basic structure of the model. Another thing that is very important that we have put in the Insighting model is stratification. We know that this pandemic is not equally affecting every age group. So we have 9 age groups for that. And also we have the provision of using this differential contacts. So the contacts between different age groups are not uniform. So we have differential contacts between different age groups. So include that in the model. And the model must have this type of properties. For example, if there are lockdowns or non-pharmaceutical interventions, this should be included in the model. So with that, the Insighting model, okay, another thing is that these infected categories are infecting the susceptible. Now, with an infectivity parameter beta, which is related to the reproduction number. With that, the Insighting model looks like this. The baseline model is taken from the Childs Mordecai et al. That came last year. And the diagram can be represented by the set of differential equations. Now, each of the compartments in the differential equation now have 9 age compartments. And these age compartments are correlated. They are correlated with a contact matrix. And the contact matrix looks like this. And at the same time, here you can see that these infected people are infecting the susceptible. But if you have good testing and quarantining measure, you are removing those people from the system. So effectively, those people are not infecting the susceptible. So we have an evolving efficiency factor, and the timeline of the quarantining and testing is represented by a parameter tau. All right. So as I said, this is the contact matrix, which we took from Prem Cook-Jit's paper. I'll be focusing my talk mainly on the first wave, which also the preprint, our preprint is all about. So the first wave can be characterized by the first detection of the case in India till 15th of February, when again we witnessed the beginning of the second wave. So the blue curve, or the blue curve, is showing you the daily number of, or smoothened daily number of cases, and the inverted orange curve is showing you the daily number of deceased people. Okay, and you can see that around middle of September we had the peak in the infected. And this is the 68 days of lockdown. This box represents the 68 days of lockdown that we had. Okay, now let's come to the data. So we have the model, we showed that what is the model. Now we have to come to the data. We try to do the data analysis with the InsiSim model. So we use two types of data. One is reported infection and reported deaths. Okay, and this is a typical example of the data plotted for Delhi. So the top one is daily infection and the bottom one is for the daily deaths. And we fit the daily numbers, not the cumulative numbers. Of course, there are outliers in the data. For example, Maharashtra reported on one day nearly minus 7,000 infected. So that means that they are trying to accumulate all the extra reports and then they subtract it. So those type of outliers you have to definitely distribute over the past few days. All right. The question is that can our model fit the data? So this plot is from our very first release of the reports from the InsiSim model. As you can see that this is a typical bell curve and it is not able to fit the data. By looking at it, you can say that. Of course, this data has a multi-peak structure, you cannot fit that. What if I tell you that I am now patching different InsiSim models and I am joining different SIR curves, all right? As you can see that this can give you a representation for the data. This can represent the data pretty well. All right. Now, this is not the end of the story. The question is that how to attempt that. Then the question boils down to the parameterization. So we use an adaptive parameterization. Here, the total timeline, as you have seen, is divided into movable windows. We break the timeline into windows. And the infectivity, the beta parameter, changes between these windows, which takes care of major changes in the dynamics. For example, complex function of the government policies, migration, stopping migration, etc., new variants, etc. These are effectively introduced in the changes in the beta parameter. We also vary the initial exposed number of people, which we do not know a priori. So this is a free parameter. And again, the tau parameter, the time scale of testing and quarantining, that is also a free parameter in our model. Then it comes down to the infection fatality ratio, okay, or the infection fatality rate. We know that if you have better infrastructure, better testing, or faster testing, you can avoid many deaths. So the infection fatality ratio over time, when you have knowledge of the pandemic or the knowledge of how to handle the pandemic, uh, the infection fatality ratio can decrease. So we are using it as a monotonic decreasing function over time that interpolates between 0.3 to 0.05%. These are supported by serosurvey and the IFR estimation since the beginning of the pandemic. And we are using two parameters for this function. Then there is a bias parameter. The bias parameter says that this is a multiplicative factor between the actual and the detected infection. This is also assumed to be decreasing over time because because you assume that your testing is going better and better, so you are reducing the bias. Then it comes to the uncertainties in the data. There can be several types of uncertainties. For example, there can be intrinsic uncertainties that can be coming from the scatters in the data because fluctuations are present in any data. So we need to simultaneously fit for the errors in the data, both infected and deaths. Then there can be uncertainties because of this undercounting of infection, because we cannot test everyone. If we test a lot of people, it will lower the test positivity, which lowers the infection undercounting and thereby the bias. So this is taken care of by our bias parameter, as I said. Then undercounting of deaths. Several sources indicate that India, many states, many districts in India are doing death undercounting because of the various reasons. And how you can understand that, you can compare with the serosurvey. I'm going to show you one plot for that. Using a comparison of our model with the serosurvey can reveal this factor. Then once you have the data and the model, then the matching comes. How are you going to match the model with the data? What is the measure of the goodness of fit? That is done by a likelihood, a mathematical construct, a statistical construct, I should say, to be more precise. So likelihood is the measure of the goodness of fit. In simple terms, it says that how likely a sample from our parameter space from that model represents the data. So once you have the likelihood is coming from calculated from both infection and the deaths. And the next step, we are going to ask that we need to sample our parameter space according to the likelihood. All right. So you need to build a code or code base. Gautam, how am I doing with time?
You have about 3 minutes more.
3 minutes more. Okay. So we have developed this code called Elixir, extended zone link 9-compartmental SR model. It is freely available in publicly available in this repository, GitLab repository. We have integrated with a Bayesian framework with a sampler. This polycode sampler is used for heavy lifting Bayesian analysis in cosmology. And using that, we sample the whole parameter space, and from the parameter space, we get the time series. Okay, now let's come back to the Delhi data. So this is the data. Now we have run our model, the incisive model, and sampled the parameter spaces. And from there, what we did is that we randomly chose 100 of the lines that are supposed to represent the data there. Okay, so these are the lines that we get. So this is how our model is describing the data. All right. So you can see that the scatters in the data are well addressed by the variations in the theory. All right. So there are lots of scatters. So you can see that the variations in the theory is taking care of the scatters. From the density of these lines, I can address what is the confidence interval, what is the 95% or 99.7% confidence interval, saying that, okay, my theory has to be between these and these values. So this is what the daily analysis looks like. And we have plotted the confidence intervals here. With that, that is how we know that, okay, our model is doing well. What we can say extra, what we can say next from that, what the data cannot say. So we can estimate the death undercounting. For example, for a given IFR, the infection fatality ratio, the actual infections can be estimated given the mortality numbers. And for certain IFRs, this estimation should match the seropositivity. And we have used the seropositivity data from this paper. Professor Rajesh Sundaresan, who is the next speaker, is also a part of this team. And so we have an estimation of this positivity, the actual infections, and from the serosurvey also, we have an estimation of the positivity. These numbers should match for a given IFR. If there is mismatch, the ratio can trace how much we are undercounting the deaths, because IFR should be consistent in across the districts in a state. All right. Now, roughly consistent, I should say. Now we perform this analysis for all districts of Karnataka, and we plot the death undercounting factor from our analysis in this colored line, colored plot. For example, this color represents the how much death undercounting is done in different districts. So if we go to the bluer colors, that means that we are doing more death undercounting. If it is more white, it is we are not doing much undercounting. Okay, compared to a Bengaluru urban as the baseline, where we assume that there is no death undercounting. Then what we do is that we population weigh the undercounting for all the districts, and that gives us the average death undercounting for Karnataka state. And that turns out to be nearly 2.2. So that means we are nearly undercounting deaths by a factor of 2. With that, we analyze the India data again, So the national data fit using the incisive model looks like this. We are fitting the daily cases and daily deaths, as I already said. As you can see that it is both the data are addressed well by our model. So this is fine, but what we can predict from here, as I said, the model should have a predicting power, right? So we can predict the actual infected from the bias, right? So here we are predicting that the end of the first wave, the India should have like 40%, 30 to 40%, or even if you talk about 2 sigma, it is nearly 20 to 50% infected. And we are undercounting cases a lot by nearly 100 times, nearly around April, which came down to nearly 20 times at the end of February this year. So the bias factor has decreased. And from there, we can also reconstruct what is the IFR evolution. So for example, we find that it is consistent, our results are consistent with 0.1% IFR. And we can also reconstruct the effective reproduction number from our time series. And this looks like the shaded lines from our analysis. And if you just use the infected data using certain other Bayesian reconstruction, independent reconstruction from this source, are plotted here in the black dots, right? So you can see that our results independently is confirming the another results from another analysis. So at the end of the first wave, we can conclude that from our analysis, we find that there are 40% infected cases in India, actual infected cases. And the results are consistent with 0.1% IFR. Cases are undercounted by a factor of 20. And urban population, which I haven't showed, but in the preprint, you can go and find the details. So urban population is significantly more infected than the rural population because of less density, obvious, and few obvious factor, less traffic, and less thereby for less contacts between people. Okay, now these are our ongoing attempts. We are trying to explore reinfection. We are trying to explore vaccination and migration effects. Although the migration effect is effectively introduced in our adaptive parameterization, we are trying to also use the available hospitalization and testing data. All what we are doing here, we are trying to basically build up a strategy to prevent further wave, if not waves. All right. So with that, I would like to stop. Thank you.
Thank you, Dheeraj. If you stop sharing and just, yeah, thanks a lot, Dheeraj. So let's go on to our next speaker, who is Rajesh Sundaresan. Rajesh is a professor at the ECE department at IISc Bangalore. He's currently at the ISI as a visiting professor. His interests are in communications, computation, control over networks, network analytics, etc. But on COVID, he's been doing really nice stuff, both with the sero surveys in Karnataka, as well as large-scale agent-based models, city-scale agent-based models that work both for Karnataka, both for the city of Bangalore, as well as for the city of Bombay. So we're looking forward to hearing from you. Go ahead, Rajesh.
Thank you. You're able to see my screen, I hope.
Yeah. Go ahead.
Thanks. Okay, so I'll speak about strategies to mitigate COVID-19 resurgence, and this will particularly be a case study of Karnataka. I would like to thank several people that were partners in this work: Shivatreya and team from ISI, Madhav Marathi and team from University of Virginia, a large group of members from IIC, and I'd like to thank some people that have funded our efforts, and my hosts at ISI and Strand Life Sciences, where I've been spending my sabbatical. So this is a picture taken from Shiva Atreya's website, which tells us about the total active cases in Karnataka. So this is the first wave in Karnataka, and this is the second wave. We know that the second wave now, after the fact that largely the driver was the Delta variant, but predicting when the next variant will come, that is next more transmissible variant will come, and what its transmissibility will be is difficult business. So what we'll try to do is study something which is actually a little more tractable, which is possibility of the next wave due to perhaps antibody waning. So here is a plot from this particular paper, Muk et al. It appeared in the Journal of Infectious Diseases, which studied a cohort of participants in a longitudinal way and called them for a second visit, third visit, and a fourth visit. You'll see here that actually there is quite a bit of variability in the neutralizing antibody levels across participants. The red one corresponds to the first level. The green ones correspond to the second level, and the blue ones correspond to the third level. But if you look at the first level and the second level, amongst participants, there is actually a significant amount of correlation between the second level and the third level, similar correlation, third level and first visit and the third visit. This is a kind of correlation. And you will also see that there is a reduction as it's given in this picture. So if this were the first level, reference level of at the first visit, at the second visit and the third visit, there is roughly a 25% reduction every 2 weeks. This is somewhat consistent across gender, across severity of the disease, and so this waning seems to be reasonably robust. So perhaps this could be used to study whether there might be a subsequent surge. And so we wanted to check whether antibody waning by itself may cause a resurgence. Variants are important to consider, but our epidemiological modeling is still work in progress to incorporate variants. But if the no variant model with antibody waning shows resurgence, we better prepare. So that's what we wanted to check. So that was the motivation for this work. So in the event there is a resurgence, what are the things that we have at our disposal? So the key instruments that we have used to manage the pandemic are vaccination, of course, large-scale mass vaccination, increase the healthcare capacity, and also non-pharmaceutical interventions like schools and college closures, home isolation, and on one side, and then lockdown on the other side, at the other extreme. Implementing these involve quite a few challenges. For example, for large-scale vaccination, we know that there is a significant supply side issue. Increasing healthcare capacity cannot be done overnight. I think many of us have had family members or friends who have struggled to get a hospital bed in the recent wave. And there is also another problem that we would like to look into, and that is heterogeneity across districts. So some districts, for example, this is heterogeneity that we observed in the serosurveillance that we had done in Karnataka in September of last year. You will see that a district like Haveri has about 8.7% seroprevalence, whereas Ballari here has about 40%. So there is significant amount of variability across the districts, and if in terms of how it has been affected. And if you take this into account, perhaps there could be better strategies that we could devise in terms of maybe we know that there are more vulnerable in this area. We might want to go and protect them. So are those good strategies? So how do we go about designing such strategies? Just to highlight a little bit more about heterogeneity, we actually ran a simulation based on when each of these districts, there are 30 districts in Karnataka, and Bangalore itself is a large entity. So we subdivided Bangalore into 9 units. That gives us about 38 units. When would the pandemic have peaked had we not taken the kind of made the kind of interventions that we did around April end? So we see here that there are a few districts which would have peaked in May. These districts from Ballari all the way to about Uttara Kannada, they would have peaked in June, and then these few districts would have peaked in July. So as you can see, there is a significant variation in the level of infection at April when we started these simulations. So there's significant heterogeneity, and that suggests that we might want to think about appropriate strategies to cover these districts given the limited amount of vaccinations that we have. So here were the questions that we wanted to address. Given the supply side pressure, will there be a third wave just from antibody waning? And if there will be, we should make preparations for that. How to allocate the limited vaccines across the districts of Karnataka? Given that we might have significant vaccination shortage, there's only this much amount of vaccination capacity per day, let us say, and a health services capacity, what should be the level of non-pharmaceutical interventions? And what about some closed loop strategies? So we had these questions, and we devised a method to study these questions. So I'll now quickly describe the methods. Largely, it goes along the lines of what Dheeraj had said in the previous talk, but it's a somewhat simpler model than what the Incisum provides. So we built an SCIRV model, so susceptible, exposed, infected, recovered, but we also included vaccinated. We also included vaccine efficacy and the delay to get this efficacy as well, a 14-day delay. Additionally, we built in this immunity waning based on one of the slides that I had shown. And there was also a vaccine supply limitation. So we brought all of these things into our model. And here are some additional things that we had taken into account. So we know that we have very many homes which are intergenerational, and therefore we brought in age stratification and age stratified contact rates. So these were additional compartments that we had introduced. We also were interested in how things would move from one district to another, because after all, we are thinking about vaccine allocation strategies across districts. We need to handle each district as a separate unit. But we brought in mobility with data that we could gather to some extent, and then brought that into the picture here. In the simulations that I will show, we actually removed the mobility, but it is a feature that we actually have brought into the picture so that when this data becomes available, we will be able to incorporate it quite easily. And we use the seroprevalence data from the first round to initialize our simulator. And as I mentioned, vaccination efficacy, 66% was the efficacy. That was because the AstraZeneca reported efficacy was 66% after 2 doses, but we sort of were optimistic and assumed that that's the efficacy after 1 dose. And additionally, I mentioned immunity waning. So with these things, we built an SCIRV model. The data sources were the state level serological survey for round 1, Karnataka daily bulletin data, which we have been scrubbing thanks to Shiva Atreya and his team, from which we got the cases, vaccinations, testing data as well, and then Karnataka demographics from the state's statistics department. So all of these have been used to build this sort of SCIRV model, but no variant modeling in today's presentations. One other thing that we wanted to do was we didn't want to overfit. There were already quite a few parameters. You can imagine that we have about 38 units for the various districts of Karnataka. Additionally, there are age stratifications as well. So what we did was we had very few parameters, which was the contact rate for the second surge. We let the simulator run, but the tuning was only on the contact rate for the second surge. And for this, what we did is we did a validation where we fit the data between the 1st of April and 22nd of April, and then looked at what kind of error we got projecting forward until 11th May. That gave us about 9% RMS root mean square error. So this is something to keep in mind. And then what we did subsequently for the simulations is we let our parameters be trained on data all the way from 1st of April to the 1st of May, and then we let it run forward. But the situation on the ground was changing quite rapidly when we were doing the simulation. There was a lockdown that was announced, and then there were some relaxations that were allowed. So what we thought would be best to do was to have several hypothetical scenarios. So this is the no lockdown scenario where mobility is not restricted. And then we uniformly brought down mobility to 1/3, 2/3 of the original level. So we reduced mobility by 1/3, which we call 1/3 MPI. And then there was a 1/2 MPI that we studied, and then a 2/3 MPI. So this is the most restrictive intervention. So I'll now present some results just to give a flavor for what to expect and also what this tool is capable of doing. So the first question is, will there be a third wave? And here you will see in this picture time on this axis and the daily cases on this axis. What we have here in red is the daily daily number of new cases. You will see also several curves. So there is the no NPI, which is the blue curve, and then 1/3 NPI, which is this dark green, and then 1/2 NPI, which is the cyan. It's kind of overwritten by the black, and then the magenta, which is the most restrictive NPI. The thing to note is that, and this is for the full state simulation, where the vaccination capacity is 1,67,000 per day. This was the number that was between the 1st and the 14th, the average number of vaccinations that we were able to give, first doses that we were able to give between the 1st and the 14th of April. So we have assumed that we'll be able to maintain that capacity. And the thing to notice here is that all these 4 curves, which are no NPI as well as the restrictions, they all have a surge sometime later in November, which is indicating that the antibody waning is putting many of these individuals that were infected back in the susceptible pool. And if there is residual infection, it will start once again. So the third wave is visible even with the strongest NPI, that's the magenta curve, with 1.67 lakh vaccinations per day. Now, we know that the peak may be advanced or delayed because during this phase, there is significant amount of stochasticity. And the height may also vary depending on the transmissibility, etc., or reinfection rates. The other thing is possible variance, which we have not taken into account. So this is without variance. The reason for this resurgence is because there is a significant fraction of susceptible population that is available for the virus to infect once again. So that's the reason. And so it is prudent to prepare for the next wave. So the seroprevalence is for round one somewhere here, and it's matched to that. The seroprevalence across Karnataka was around 27%. So that's what we have in this picture here as well. Okay, there is a possibility to match to the second serosurveillance data that's around here. And so this is something that we should do. We have the data, and we have visually inspected it, but it's not been calibrated to that yet. So the next question that we wanted to address is allocation across districts. This is important because there are some vulnerable districts and some where the infection has possibly raged, and how do we allocate vaccinations across these? So assuming a 1.67 lakh capacity per day vaccination capacity, what we have done is studied 3 different policies. So here you allocate vaccinations to districts in proportion to population. Here in proportion to the predicted seroprevalence around February. And this one would be matched to the incidence rate during the early part of April, because if there was a surge in a particular district, then you might want to go and vaccinate there a little more aggressively, a sort of ring vaccination, so that things don't spread. What works better? In the short term, it turns out that if you compare the reds, this one is marginally better. So ring vaccination is marginally better. But if you look at the long term, July as well as August, it turns out that vaccination allocation in proportion to the population is best for the state as a whole. And so the suggestion is to allocate vaccinations in proportion to the population, keeping the longer term perspective in mind. I think this is a valid insight that we can share with people that are managing the pandemic, and it's something that can inform the logistics related to vaccine distribution. So that's as far as this question is concerned. The next one is we don't have enough vaccines. So what level of NPI might we have to use in order to manage the pandemic? So very quickly, so let us assume that the capacity is, instead of 1.672 lakh doses per day this time, and we can, our health services capacity is capable of providing good care so long as we have under 1 lakh active cases. If that were the case, then during the May timeframe, we would have struggled. We would have had to have a very stringent lockdown, which is what we had, and vaccinate aggressively, but we didn't have enough vaccines during that period. But things do ease, or did ease in the early part of June. So at 2 lakh vaccination, so what you should look at here is this vertical line corresponds to, on the abscissa, one third is somewhere here, 1/2 is here, and 2/3 is here. And all these ISO lines indicate what is the active cases that will come when we use a particular level of non-pharmaceutical intervention and a certain level of vaccinations per day. So if we do 2 lakh vaccinations per day, and let's say we can handle about 1 lakh active cases, then we should look at the intersection of this 2 lakh horizontal line and the ISO line corresponding to 1 lakh. So that suggests that we should have a little stronger than 1/2 NPI during this period. But that can be relaxed during the June timeframe for the same 2 lakh and 2 lakh vaccinations per day and 1 lakh active cases capacity. We could do with a 1/3 NPI, and this can be relaxed a little further now. We can have maybe a 1/6 NPI. But the interesting thing is that if we want, let us say, a lower NPI, this picture also suggests that you must vaccinate way beyond. So let's say we want only 1/6 NPI, then you must vaccinate significantly. On this you're outside the chart here for the 1 lakh ISO line. You should vaccinate beyond 4 lakh. So there are these kinds of things that you can interpret. It also suggests that perhaps we can react to this in closed loop.
Rajesh, can we wind up now?
All right, yeah. So I will skip this closed loop. And what I will do is quickly come to the last slide. So there is a third way, even with the most stringent NPI that we studied, and 1.67 lakh vaccinations per day. Vaccination allocation can be done in proportion to the population. This is a useful finding. we have provided a chart to determine the MPI level for a given vaccination capacity and health services capacity, and can use this to come up with closed loop strategies, which I did not discuss, but it's something that is quite clear from the pictures that I showed. Thank you. Sorry.
Thanks, Rajesh. Thanks, Rajesh. No problem. Can you stop sharing? Yeah, thanks. So thanks. We'll now move on to Chitra. So Chitra Pattabhiraman is a virologist and molecular biologist who uses genomic tools, sequencing to identify pathogens and brain infection. She's been working very hard on COVID-19 sequencing in the Bangalore, Karnataka area and more generally. She's an India Alliance early career fellow at NIMHANS in Bangalore. And she says she's interested in all things viral. So go ahead.
Hi, thanks Gautam for that introduction. Can you see my screen?
Yes.
Okay, great. So basically, I'm just, I can see that I have a spelling mistake in my title, but basically what I'm going to do is talk about genomic epidemiology of SARS-CoV-2 in India, and basically take you through what we have learned from genomics and lead up to Vinod Scaria's talk, in which he's going to talk about continued genomic surveillance and the importance of that and some of the programs that he's helping set up. So the idea is to build up to that. As all of you know, this is a small world for a virus. In some sense, we always knew that. From Uma's talk, you already know that we are at that place where we expect pathogens to emerge, and we are such a connected world that it's very likely that once they emerge, they are going to spread. So this is the coronavirus, and by now it's everywhere, and as you know, this is like a global pandemic. So the genome of SARS-CoV-2, you can think of it as a long thread of around 30,000 base pairs, and each of these bases can be AUGC, because it's an RNA virus. And this is how the genome looks. Basically, there are untranslated bits in the beginning and the end, and it is divided into this coding regions, which make different kinds of proteins. The most important of those is the spike, because it helps the virus sort of sit on the cells and get inside the cells. So, and that is what gives the coronavirus a spiky appearance. So we can represent the genome sort of linearly like this, different parts of the genome, and that's what I will be talking about a little bit. So this is just to introduce that. So this virus, because it's an RNA virus, mutates at a certain rate, and very roughly this is about, it picks up a mutation in 1 to 2 weeks. So basically in this—
just a quick interruption. I think we are not seeing the slide progression. So perhaps you could share slides once again.
Hi, are they visible now?
Can you go to the next one? We'll just check.
This.
Yeah, it's working.
Okay, so I'm going to use this to move and not have a slideshow mode, if that's okay with everyone. Yeah, so basically this is a 30,000. Is this okay?
Yes, that's fine.
Okay. So basically, the genome is about 30,000 base pairs long, and we can represent it linearly like this. And this is sort of where the spike is situated. Because it's an RNA virus, it mutates at roughly about 1 mutation every 2 weeks. And this can be anywhere in the genome, right? And so basically, all genomes, starting from the SARS-CoV-2 that was circulating in Wuhan, all of them are variants of SARS-CoV-2. And it is clear now, after looking at in the beginning of the outbreak. So in the beginning, there were only 11 mutations that we were seeing again and again. So there were very few. But over time, mutations have accumulated in the genome. Like Vidita mentioned, that they're tending to converge in certain places in the genome. So you can see the counts, that is a diversity count at a particular codon and across the genome of the virus. And you can see that it's clustering into some places. So there are hotspots and cold spots for this virus. And I think Vinod will also talk about some of the convergence that we are seeing with respect to the variants of concern. So basically for SARS-CoV-2, what are these lineages that people talk about? So basically a lineage of SARS-CoV-2 is they are related viruses. So they are clustering by phylogeny. That means evolutionarily, we expect them to have a relationship. Then they're associated with an epidemiological event. So basically we are calling something a lineage when we know something about their origin, that is something about the first time they were sequenced, that is. So here, for example, these pink ones, they are sisters, they're sister clades, and they seem to have come from a same place here in the UK. They were reported first, so they might form a separate lineage. And the Pango classification, which we use, it actually classifies viruses as the parental virus as A, and then B, B.1, and so on, and where B.1 is a daughter of B. And if you go along in this scheme, you'll see that after 4, they actually switch the alphabet. So a daughter of B.1.1.1 is no longer going to be called B, it will be called some other letter. So that's how you get all these different B.1, B.6, and so on. And what this does, what this allows us to do, this kind of nomenclature, is that it allows you to already know that B.1.1.1 is actually a daughter of B.1.1. And so they are related to each other. There are over 2.1 million sequences, SARS-CoV-2 sequences in the GISAID database. This is a really huge number for any virus. And so a lot of tools are being built to even use this data well. There are more than 800 global lineages. So what has happened in India over time? So this is looking at SARS-CoV-2 lineages using the Pango scheme. And you can see initially in March 2020, we had a few dominant lineages, which pretty much died down by July 2020. And then this lineage here, B.1, came up, and all subsequent lineages all across the world are daughters of this lineage, B.1 and then B.1.1. And they sort of took over. So all of this space is daughters of this lineage. And you can see that for a long time then, we had a few lineages, but not very dominant ones. And these were actually different in different parts of India, if you break down the data. Then in around November, December, you have this variant, which was first reported in the UK, also known as Alpha, B.1.1.7 come up around here, mostly introduced by travel at this time. Then by March, you can see it had come in, like there are lots more of this, suggesting that it had come into community. And we also know that from other data that we've analyzed. At the same time, you start seeing this B.1.617 come up. And now pretty much daughters of this lineage are dominant. They've taken over completely. So this is the Delta variant, right? So I'm going to tell you about the first initial cases that we did sequencing and tried to understand what was happening in the state of Karnataka again from March to May 2020. There were 1,500 cases roughly in this time. So this is very early. And what you see here is the distribution of cases across the different districts of Karnataka, which have also been very nicely introduced before. So what you see here is Bangalore urban. So even by that time, Bangalore urban had the maximum number of cases. In green are the number of samples that were tested at NIMHANS, because NIMHANS is where I work, and that's where this study was done. So, we got representations from many different districts, and we sequenced from within what we got. So what we sequenced was a reasonably high number of what we got, and I'll tell you a little bit like about the numbers also. So basically, this was the sort of profile of what we sequenced. So the state had about 1,578 cases. Our lab got 369. We sequenced almost 1 in 4 of them. And you can see that the age distribution that the state had at that time was skewed towards younger people. And we sort of had that in our representation as well. We knew at that time that there were more men than women who were infected. And then, so that was also sort of there in our data. And between asymptomatic and symptomatic people, we tried to sequence symptomatic people, but we got very few of them in NIMHANS. NIMHANS was a center that was actually testing asymptomatic people more often, so contacts of people and so on. So we had contact tracing data from Karnataka, and so we knew about the travel histories of all the people that we sequenced. So there were 700 cases with known contacts, and there were some more cases with known travel histories, but there were quite a few already by that time where we couldn't assign a source. And so in our study, one of the things that we tried to do was, for this proportion that we could sequence, we tried to assign possible sources to that. And so what we had was we had contact tracing information, as I mentioned. So we did cluster analysis. So there were 104 contact clusters in the state at this time. We sequenced only 17 of them, but we captured about 1/3 of all the clusters with more than 5 people. So I think it was pretty reasonable, the representation that we got. So this is what we had. Uh, so we got 7 lineages of SARS-CoV-2 from our sequencing, about 101 cases. And most of them were B.6. This is not surprising because it is clear now that B.6 was the dominant lineage at that time. And there was a super spreader event associated with that as well. And all of these were the other lineages in Karnataka. Many of them, the introductions were like international travel, known history of international travel, and so on. So basically, there were multiple introductions of SARS-CoV-2 into Karnataka. This is the phylogenetic map, which is basically phylogeny, which is just telling us that they were quite distinct from each other. At the time when we were doing this, it wasn't clear whether the lineage was the best way to look at this relationship. And what we found is when we did this sort of network building based on those contact clusters, we found that most of the time within the contact clusters, so 17 large clusters and individuals with no known contacts, when we put them together, we could see that mostly in a cluster, we had a single lineage within that cluster. We also looked at this data based on asymptomatic and symptomatic people. So based on that, we found that mostly you had a symptomatic person at the center with the maximum number of contacts. And at this time, because there were lockdowns and so on, the clusters were contained within districts. There were very few clusters with more than one district. And this helped us sort of assign which were the clusters that were breaking. So we could see a lot of B.6 already not being in clusters and so on. So what we did in that study was basically we linked epidemiological data with genomic data. And we think that this kind of approach, doing it in real time, is necessary for an effective outbreak response. And what we figured out from that was there were multiple introductions of the virus into the state. There was continued importation by domestic travel from other states, even during lockdown. Symptomatic individuals were spread across multiple lineages. So there was at that time no variant that seemed to be causing more severe disease than others. And so that is what our data showed us. So later, between November and Jan, we sequenced again. This time because the variant from the UK had been described, and we wanted to know about its introduction and spread. And what we found was that in 73 imported cases in this time period, there were quite a large fraction that were of the alpha. And but locally, we had a completely different profile. So if you looked at Bangalore at that time, this is what we had circulating. And this changed very quickly, as you know. So this is sequencing efforts through the INSACOG initiative as well, and data from both NCPS and NIMHANS combined. And what you can see is that over time, you can see that the lineages have changed as a fraction of the total number of sequenced SARS-CoV-2 genomes. We had initially mostly this B.36.29 for a long time as the dominant lineage. Then we have the introduction of B.1.1.7 or the alpha variant, and that sort of got introduced into community somewhere in March. Sorry about that. And then slowly we see the delta coming in and taking over. This is more clear if you look at this as a percentage breakdown, and you can see the shifts in the lineages. So what that roughly tells us is that continuous genomic surveillance locally is necessary for detection of variants and variants of concern, which hopefully Vinod will talk about. And variants with mutations associated with immune escape and reinfection are already circulating in India. And mutations in the sequence of the spike protein, which I didn't talk about actually, can arise in any lineage. It is not restricted only to certain lineages. We actually see it in many different. So it could be, some of what we are seeing could be the opportunity to expand, could be part of the problem, not just the property of the variant. right? So it is important to track low frequency mutations occurring in a lineage at a local level. And we need to combine genomic information with epidemiological data rapidly, and that might guide public health measures. So all of this work has been done by lots of people, many different teams. So these are the people doing the actual sequencing in NIMHANS. Of course, we wouldn't have these samples if we didn't have a diagnostic lab within NIMHANS. So there are a lot of people working in that. Dr. Ravi is now the nodal officer for genomic confirmation of SARS-CoV-2. So we have a lot of, we work with the government quite a lot, both to provide them with information as well as getting information that we can combine with sequencing data. A lot of the network analysis that I showed you, it was done by Farad, who's a data scientist at InMobi, but helped us with this. The IGIB team has consistently helped us throughout, and some of the INSACOG data science data analysis team as well. And we need to thank everyone who's submitting data to GISAID, otherwise we wouldn't be able to do this. Thank you.
Thank you, Chitra. Thanks. If you just, yeah, thank you so much. So now we go on to the last speaker in the session. We'll go on a little bit more so that we have time for the questions that people will ask. So the last speaker here is Vinod Scaria. He's a researcher who's pioneering in precision medicine and clinical genomics. He's currently at CSR's IGIB and also at IIIT in Delhi. And among many other things, he's a pioneer in clinical genomics in India and is also interested in rare genomic diseases, where again, he's done very important work. Vinod, go ahead.
Thank you, Gautam, for the nice introduction. Without losing time, let me get into my topic. So over the next 15 to 20 minutes, let me talk about how genomic surveillance has been used to impact public health. I'll discuss a few examples from the early surveillance across the states with some emphasis on Kerala and, of course, the way forward. For students out here, I think this is the right time to also discuss a bit of history. When you talk about microbiology, we pretty much talk about microscopy, and the father of modern microscopy is nobody other than Anthony von Leeuwenhoek. Now, 300 years after the invention of the microscope, we do have a much better microscope, and fortunately what we call as a genoscope. Conceptually, they are very different. In the first form, they look at the size and the form of the organism, and in the genoscope, we typically look at the genetic material of the organism. Now, the tools to look at such large-scale techniques did not exist before the COVID pandemic. And one of the major changes or the paradigm shifts that has happened during the COVID pandemic is the implementation of tools of extremely large throughput that can enable genomic surveillance. I mean, it's not that genomics did not happen before the pandemic, but it had never happened at the scale and throughput that it has happened in the last few months. And one of these techniques for high throughput sequencing is what we call as a COVID-seq. Unfortunately, not many people know that the first clinical validation for this high throughput sequencing approach, which is now followed across the world, happened in India in the lab of my colleague, Dr. Sridhar Chalsupu. Now, this approach is quite modular in the sense that it can scale from anything from 96 samples to around 3,000 samples in around 24 hours. And this throughput can actually give you a lot of advantages, and that's the advantages which allowed us to look at a few states across India in early pandemic. And this is sometime mid last year, where we worked with a large number of state governments in trying to understand the genetic landscape of SARS-CoV-2 in those states. This includes mostly the southern states of Tamil Nadu, Kerala, Karnataka, Andhra Pradesh, Odisha, UP, as well as Rajasthan. And of course, there's a lot that we did and a lot that we could discover in terms of new lineages, etc., etc. But what's more important in terms of conceptual understanding is that when we do genomes over time or genomes spanning multiple time points, what we're trying to understand is essentially genetic mutations. Now, genetic mutations in SARS-CoV-2 occur at a very, very constant rate, and I'm sure Chitra did mention it quite elaborately. And this constant rate of new mutation occur in the background of an existing mutation. In other words, you can sort of trace the origin or the genetic lineage of a particular virus just by looking at the genetic lineage of the genetic material of the virus, and also understanding what were the previous genetic lineages which are predominant in that region. Now, typically, since the mutations occur at a very constant rate, at any point in time, you expect to have equal proportions of all lineages, but that's not what you generally see. You generally see a quite large distribution of a particular lineage. In this particular case, there is, for example, mentioned 1, 2, 4, which occurred in the background of 1, 2, which nevertheless occurred in the background of 1, and so on and so forth. Now, these are what we call as emergence events, and typically emergence events are caused by two independent factors. One is a human factor, like, for example, a super spreader event, or secondly, it could also be because of a highly transmissible virus. Now, when we look at emergence events, we essentially look at a large proportion of a particular lineage of the virus. In this particular case, let's call it for the namesake 1 to 4, in comparison to the other lineages which are prevalent across. Now, one of these lineages which we early identified in April and May of this pandemic was this very unique and distinct lineage, which is colored in violet in here, which we call as the A3I. I to signify this was a lineage. This is almost exclusively found in India, but of course in small numbers in many other countries. And A3I to also signify that it was an offshoot for the A3 lineage. The A3 lineage was essentially the old next strain lineage classification for sequences out of Iran. Now, what we could identify as this particular genetic lineage came from a super spreader event. You can trace back the origin by the kind of tracing that I sort of mentioned before. And this was prevalent across many states in the country, the largest proportions from Delhi and states like Telangana and Andhra Pradesh, where there were a minority population. So this is one of the applications of doing genomics, to identify very specific lineages, not just identify lineages, but also understand their origin and their spread across. And as you see here, over time, this particular lineage ceased to exist because it was overwhelmed by another lineage, what we call as the A2A, which contained the D614G genetic mutation in the spike protein. The other application of looking at genome sequences from across the country is to be able to do what we call as molecular contact tracing. Now, essentially, the concept of this is very simple, and I sort of exemplified in the earlier slide. If you know a particular lineage is 1 to 4, then you know it happened in the background of 1, 2, which nevertheless happened in the background of 1, and so on and so forth. So in other words, looking at lineages or isolates from across the country, you would be able to understand how did it move between cities of the country. And one of the applications of this was in the sequences from Kerala. And Kerala, as you know, has quite large population who live abroad and travel very frequently to the state. Now, Kerala had a very draconian kind of surveillance measure where everybody who were traveling from abroad had to go to a 21-day quarantine, which was in many ways not widely appreciated. I'm sorry for that. Not widely appreciated by the people out there. Now, genomic surveillance essentially showed that the predominant lineages of SARS-CoV-2 in Kerala came from largely 3 clusters, and none of these clusters had an international origin. In other words, this is an application of genomics where you can sort of validate the policy of quarantine, which essentially sort of says that the largest proportion of genomes in the state of Kerala did not come from international travelers, but actually came from travelers who stepped across state boundaries. Now, after this initial pilot experiment, Kerala initiated a very, very systematic program encompassing 12 medical colleges, the state public health laboratory, university departments from the Inter-University State Center for Biomedical Research, as well as from the Central University, as well as the 14 district surveillance units, which were part of this very large program. And the idea of this program was to do a systematic genomic surveillance covering every single district. Kerala has 14 districts, a systematic survey which encompasses 100 samples every month from any of these districts. Now, briefly speaking, and compressing all the data for the last 6 months, Kerala has seen 3 lineages which came in waves. One is the D1.560, which came early during the Jan and Feb months, which was followed by the B1.1.7 or alpha, and which was later followed by delta in close succession. So in other words, not just understanding the spread of the virus over time, this can also enable implementation of policies. One of these examples is exemplified in here, when a significant large proportion of the virus of alpha and delta lineages was seen. It is very well understood that this is of very large transmissibility, and therefore Kerala implemented a policy of double masking and as well as self-lockdown, which was probably one of the first in this country to implement a policy of double masking in public. And of course, as the cases rose and B1.617 or delta had been increasing, the state went into a lockdown. And the effects of this early interventions of policy interventions are actually exemplified in here, in that probably of the major metropolitan cities in the country, Trivandrum has been one of the metropolitan cities which has a negative excess deaths even during the second wave of COVID. Now, how do we go from here? And what we really understand about SARS-CoV-2 is that this virus mutates in a very stepwise fashion. Or in other words, there are lineages which emerge from pre-existing lineages by acquisition of very specific genetic mutations. And over time, these genetic mutations have recurred in multiple different lineages. And largely speaking, this confers two key characteristics to the virus: either increase the transmissibility or increase the immune escape property, whereby it can evade the immune response caused by a few or many of the antibodies. Now, in terms of a landscape of how the virus is emerging, the virus has taken multiple different steps, either moving horizontally up in terms of increasing the transmissibility, or stepping sidewise to increase the immune escape. Or in other words, by this stepwise process, we have pretty much reached to a point where we have delta, which is today the most predominant lineage across India and in many regions of Southeast Asia, and of course in many regions of the world, including UK and very recently the United States. Now, the real big question is how and where will the virus move next? Is it going to move towards being more transmissible, or is it going to move towards being more immune escape? And it's of great importance to understand this and sequence in a very systematic way. And this is one slide that I would try to show to convince you how important is it to sequence in a very systematic way and in high depth. And what you see here is essentially delta and kappa. And what you can sort of, from the sequences which are available out here, what we can sort of do is to build what we call as the most common ancestor using computational tools and looking at the divergence rate of the virus. And what you see here is that delta and kappa originated in the previous peak sometime in the period from July to September. Or in other words, what you need to sequence and when you need to sequence is essentially when you have very low number of cases. That is where a new lurking variant would come and hit you, and at a point of time create a big wave out here. The point is not to sequence at the peak of the wave, because pretty much what you all sequence at the peak of the wave is going to be a unique isolate, which is what would have emerged. But what's really important in preventing the waves from coming up is essentially to sequence in great depth and in a consistent fashion in a period in time when there are little or no cases. And this is also important in terms of understanding the evolutionary history of the virus, because the spike protein is the exposed protein of SARS-CoV-2, which incidentally is also the protein which it uses to latch to the human receptor. And in fact, almost all monoclonal as well as polyclonal antibodies also target the specific region of the SARS-CoV-2 spike protein. In other words, there are structural constraints on the spike protein. It is not that there are infinite number of possibilities that the spike protein can emerge. It is at the cost of one another. So in other words, a mutation that can increase the transmissibility can possibly reduce its resistance to antibodies and vice versa. In other words, what we can also fairly say is that the genetic landscape of SARS-CoV-2 mutations is pretty much finite, and we pretty much would know almost all possible genetic mutations which can happen on this spike protein. In a state like Kerala, how does it work, and how do we move forward? And one of the approaches the state has taken is to actually fine map these outbreaks by also involving local self-governments. And as you would have also seen in news, that's probably one of the first states to implement a lockdown policy at a micro level involving the local self-governments. So the idea of and the principle of genomic surveillance as the cases come down is to report abnormalities, which includes vaccine breakthrough infections, severe COVID in the young, local outbreaks, as well as infection and forward transmission in inbound travelers, and actually use genomic surveillance using whole genome sequencing to identify variants and mop them up using targeted sequencing, using Sanger or capillary sequencing based assays. And I think I'll stop there and take questions. But before that, let me acknowledge a lot of people who made this possible. A lot of students in the lab, fantastic collaborators, including Dr. Sridhar, who's my close colleague and collaborator for almost a decade and a half. Fantastic researchers and clinicians who are part of the multiple collaborative networks. And of course, funding from CSR for the early part of the program and the Kerala Surveillance Program and further from INSACOG. And moving forward, of course, community participation in all of these is extremely important. And that's how we have also been coordinating one of the initiatives to involve a large number of public resources, including students and learned people in the community, to build the next generation of surveillance methodologies. Thank you very much.
Thank you so much, Vinod. It's a great talk. So if you stop sharing, we can just go to questions right now. We don't have too much time. We've gone a little bit over, but we do have time for at least the 5 or 10 questions that we've had. So let me just go in sequence. And so there's a question for you, Uma, from Anirban, which says, when does a spillover happen? I mean, if bats live in populated areas, but there is no interaction with human population, what are the chances of spillover? When do you expect spillovers to happen?
So thanks, Anirban, for that question. So I guess, spillover may be happening between bats within caves, right? Between species of bats. If there are different species of bats that live in a cave, that is also spillover. Spillover need not refer only to a spillover to humans. But yes, a spillover from bats to humans would occur if there's some interface between bats and humans, depending on the virus. For example, with paramyxoviruses like Nipah or even Ebola, there would need to be some fluid exchange, something the bat drinks or eats, which humans are in contact with, some fluid. Or it could be, for example, with coronaviruses, it could be, as we all know, other modes of transmission. So yes, spillover would happen to humans only if there are strong interfaces with bats. But we have to remember that bats are actually everywhere. I'm sure you see them if you walk out in the evening, beautiful black, silhouettes in the sky. Those are often flying foxes, and they have large roosts. They also provide many ecosystem services like pollination and insect control. So yeah, sorry, I took a bit long. I know we don't have much time. I don't want to hog the time.
Okay, so let me just cycle through. Let me ask a question to Vinita. Question is, what is the general probability of a coronavirus variant to transform in such a way in a short time to increase the binding affinity to human cells, to the ACE2 receptor? Vinita, I hope you're there.
I think, thank you, Gautam. I think we know the answer that in his presentation just now, that yes, there will be cumulative mutations and sort of it will go on. And as far as the spike protein and receptor binding domain is concerned, there will be certain mutations, but one can be at the cost of the other, is what he was saying, that there might be mutations which will increase binding, but they may actually be less virulent because they might help in survival of the species in the sense of viral species. So there isn't, I don't think, a single mutation which will really make radical change in the virus is likely to happen. And most mutations which we do not see are happening because there are, with every replication, there is mutation and changes in the nucleotides, but most of them do not survive. And hence we tend to see only those which survive and which have possibly some survival benefit. So answer is it's going to be a slow process and not a major jump at one time.
Okay, thanks, Anita. Let me give you another question right now from Anna Sharma. She asks, what is the relative importance of spike protein mutation versus end protein mutation?
Okay, sure. So again, structural biologists would know this better, but from what I understand, nucleoproteins will not affect ACE receptor binding so much as the spike protein mutations would. But where the N protein mutations would matter is from the rest of the viral genome, all the proteins that are part of the virus, the peptides derived from such proteins will contribute to so-called T-cell responses. So CD4 and CD8 T-cell epitopes, about which I did not talk a lot at all, those will be there, and changes in nucleoprotein may result into some changes in the CD4, CD8 epitopes, which might matter. But essentially, it is not the case that every mutation would matter in terms of global immune response.
Thanks, Sunita. So here's a question for Chitra. This is from Vishwam Shankaran. He says, is there a way for methods in wastewater epidemiology to be coupled with genomic surveillance to get more diverse sampling for variants?
Hi, I think Uma is actually a better place to answer this question, because this is something that is now being tried that is getting integrated into genomic surveillance, environmental surveillance. So I know that CCMB has been doing some of this, and NRL in Pune as well. So, and as well as some researchers in Stanford have been putting together a protocol for doing PCR-based detection of sewage. The problem there is nucleic acid integrity. And getting that nicely out of the samples, because there is fecal shedding. So we do expect it there, and probably we expect it earlier, given that people don't want to get tested. We might be able to circumvent that. So yes, this should, is being done. Thanks.
I'll ask Uma if she has anything to add to that in general.
Is it worth putting NCL data on it? I have just heard that data yesterday.
Yes. So there are several, like Vinita pointed out, NCL Pune is also, Mahesh Dharne is also doing this. Many researchers around the world are doing this. Just conceptually, if there are multiple lineages circulating, which are all going to mix up in the sewage, right? And you're going to take one sewage sample and sequence something from it. And that's all, like Chitra said, it's 30,000 letters, but they're all torn up. So what's going to be the challenge is to infer the unique lineages, correctly, and not mix up and match up things incorrectly. So yeah, this is going to be a challenge, but maybe some combination of approaches will have to be used. Thank you.
Thanks, Uma. So here's a question for Vinod. And this is from Vishnu Shankaran again, saying, given the lack of resources to sequence large numbers of samples in India, is there a way to be smart about sampling to screen for new variants, especially in advance of a big third wave potentially?
Yeah, the first answer to the question is that there is no lack of resources whatsoever to sequence samples. And I think we have more than enough capacity to sequence as much of the scale that a state like Kerala or Maharashtra is doing at the moment. The second point is, of course, to prioritize what point in time should you sequence, right? In many ways, it is a human mindset that when the wave comes, let's go back and sequence. That's in many ways going to be futile, because most of these variants would be lurking and would have already developed at a point of time way before a wave has hit. The wave is essentially a manifestation of a lurking variant which has emerged. Now, that is not going to give you any information about that is valuable to intervene, because once a wave has hit, that is pretty much when you know that you have to go into a lockdown. The really important period in time is during the wave and after the wave, rather, when the cases are really low. And this is in many ways logical, because the number of mutations that the virus would explore becomes so large when there's a wave, because there are a lot of people who are infected and therefore a lot of mutations. Most of the people in the community would be immune, and therefore this emerging or the emergent variant would be lurking well below the surface, transmitting in very low numbers, right? And that's essentially when you need to capture them and when you need to sequence and figure that out, because that's essentially the time for intervention. So if you know that there is a suspicious variant with a set of features, either in terms of variants which are associated with increased transmissibility or variants which are associated with immune escape, when they come up, they need to be, handled at that point in time, which is more economical, right? Rather than waiting till it becomes 50 or 60%, right? And when you realize that point in time, there is not much time left to actually intervene.
Can I just ask sort of a follow-up on that? Just in terms of numbers, I thought INSACOG currently is doing 0.1% or something. You know the numbers better than I do. Initial idea was to do 5% of submitted samples, but I think they're still at least about one order of magnitude less than that. So what is the context of your statement saying there is enough sequencing capacity? Is that in the sort of private sector that
yeah, I will not talk about INSACOG. Probably Vinita would be the right person to talk about it. but what I can say is, an average sequencer of the smallest throughput can actually churn out something like 96 samples every 2 days, right? So the throughput is pretty much not an issue because we're talking about around 600 odd districts, right? So that average turns out to be around 60,000 odd sequences a month, right? That's pretty much, much lesser than the established capacity for sequencing in the country, if you look at it that way, right? Of course, there are logistics issues which I'm not going to get to.
Okay, fine. Here's a question for Uma again. This is from Roni saying, can you argue that the immune system of the Northeast Indian population is well equipped to handle spillover effects?
$1 million question. I don't know the answer. And in fact, I've met with Vinita to ask her advice on this. There's definitely been exposure for long term, for a longish period of time. And this is something which these are the kinds of things which would be really important to explore with an integrative approach. Clearly, an ecologist like myself may not be able to explore these. And that's why One Health really depends on intersectoral collaboration. And I hope that, which generally hasn't happened so much in India. So I hope that going forward, we'll be able to encourage such programs. Thank you.
One more question in your direction. So this is from Prajwal, who says, how much higher was the measured exposure to the rabies-producing Lisa virus among bats studied in Nagaland compared to the average across domesticated animals in India?
So, these serology assays are not so good. They're not so quantitative, at least as to the best of my understanding. The assay we are using is a multiplex serology assay, a Luminex assay, which actually looks at multiple tests at one time. And so it's kind of a probabilistic inference of positivity, and there could be cross reactivity as well across viral families. So I'm not sure we can really get to that level of detail. But you're right, Prajwal, looking at livestock would be very important. In the Northeast, there's not that much livestock. There is, but not like UP or some other parts of the country. There are pigs and there are things like Mithun and so on. But yes, that component is important to integrate as well. Thank you.
So this goes to Vinita. So what's the difference between antibodies naturally produced by the body in response to COVID-19 and antibodies produced by a vaccine?
No difference as far as I can see, because ultimately the only major difference is when it is a natural infection, all components of the virus are presented to the body's immune system, and hence there will be antibodies also against, say, N protein and other proteins which get expressed in the virus during the course of infection and replication. As against that, most of the vaccines, except for, of course, Covaxin, is everything is practically based on spike protein or the receptor binding domain. So in that sense, anti-vaccine and antibodies generated by vaccine are more focused on those parts of the virus, and the parts were chosen primarily because spike protein is very, very crucial in binding to S2 receptor and hence gaining entry. So if there is an spike protein deficient virus, it will probably be completely useless. In that sense, antibodies and T-cell responses against spike are the most important. That's the only difference. But if you look at anti-spike antibodies post-infection versus anti-spike antibodies post-vaccination, there isn't anything major that one can say would be grossly different.
Again, a sort of follow-up question on that. Why in that case would whole inactivated viruses not be a better option? Because there's just many more epitopes that are presented. And if you have immune escape connected to the spike, to the spike, and you can, in a sense, get around that if there are other parts of the virus that you recognize.
No, not necessarily true. I mean, as I said, spike is indispensable, or RBD is indispensable as far as entry is concerned. And if there is no entry, there is nothing further. So in that sense, that basic interaction has to be curtailed, inhibited, whichever way one would like to call it. So that in that sense, antibodies against RBD, receptor binding domain, or the spike protein are really, really important. And it is true that there will be diversity of antibodies which will be generated by the Covaxin, but it's also a question of how much of, say, how much of spike protein is part of the vaccine, as against if you take Covishield, since that's the other vaccine available, how much of protein gets, how much of spike protein gets generated from Covishield. So depending on how much is the component, the immune response would be somewhat akin to larger amount of protein, larger response, smaller amount of protein, smaller response. So those are only restricted to antibodies. Apart from that, there is also an issue that adenovirus-based DNA, RNA vaccines, whatever that we have, do trigger what I called as cytotoxic T-cell response, which is very crucial for the next round of protection, meaning immune memory generation for viral infection. And that is not very readily triggered by a killed virus. So there are differences, there are advantages and disadvantages, but one has to take what one gets.
Okay, so now we're running out of questions at this point. If I sort of look at everything that we have so far, let me just ask a couple more. A lot of them are coming your way, Vinita.
There was one question on childhood something.
Yeah, that's correct. So let me just locate that and then what's the rationale behind the news that says the next wave will affect children more? Is it because next category of, in quotes, easy hosts for the virus will be children, or is there some other reason, or is there no reason at all?
Well, even today, we don't have data, but if you look at carefully done serological surveys where children were included, the antibody, meaning presence of antibody in children is also it has been detected. So even if we say that children are not affected as much, the infection has reached children. Only thing is we do not have good data to show it on a nationwide scale. That's one point. So it is not like today there are no infections for the 0 to 18 age group, and tomorrow they will be more. They will be relatively more at some point in tomorrow, and that depends on 2-3 different factors. And first factor is if the third wave, so-called third waves, people have been predicting now based on the model as November, December of this year. But until then, if the vaccine coverage is very extensive and very good, then you can imagine that most of the adults above the age of 18 years would be vaccinated, and hence more or less, I'm not saying 100%, but certainly would be more protected than children who as a group. As a result, the only major susceptible population remaining significant numbers would be children, and hence children would get infected, not necessarily because they are more susceptible or anything of the kind. It's also possible that the virus may change further, and Vinod and others might be able to predict it a little bit. How good the prediction would be, only virus can tell when it comes out with another layer of mutation. But at the same time, that also might change. So if there is a significant change, people who got vaccination or infection very early on in the epidemic, say last March, whether their antibody responses are long lasting to give them protection if they have not been vaccinated, and so on and so forth. I know I keep saying that modelers don't necessarily take all these variables into account, and Gautam and I have had probably 20 years of conversation on this. But again, the same point I'm making, that there are so many variables which need to be accounted for, that a simple answer is not a possibility. But as I said, children will be more highly targeted in terms of proportion later the third wave is.
Okay, so let's just finish up with this last question. I'm going to pass it on to Chitra, but I think both Vinod and Vinita may have things to say about this. Question: are there quicker protocols to determine if a detected variant is antibody escaping or more infectious, given the lag in communicating this anyway to policymakers? How much time do you need to tell that something is some new variant that you haven't seen before, in some mutation in some distinct site that hasn't been, it may be more substantially more immune escaping?
So one is that we have a catalog of such mutations that allow, like we have an experimental catalog based on virus that has been grown with the pressure of an antibody and the mutations that it has picked up. So this is a library to compare the mutations that we see in real life against. So I think that the initial 501Y mutation was picked up like that, and many of the others that we're seeing now were already cataloged as possible immune escape mutations. Then of course we know that there are structural constraints, but that also that it is that the spike is more plastic than we had anticipated. It can tolerate mutations more. I'll just say that much and pass it off. So there are methods and people have developed techniques, but you have to grow the virus for that. I think you have to grow the circulating strains. So there is going to be a delay. I'll pass it off to the other two. Thanks.
Vinita, you want to have a try? And then we'll go to Vinod.
Okay. The minimum time based on whatever people who can grow virus after diagnosis, or after the sequencing is done, is minimum of 3 weeks is what people have been saying. So you share the same transport fluid, viral transport fluid, and from that grow the virus and grow it in sufficient number. Two kinds of testing needs to be done. One is whether the virus, whether the antibodies present in the population, either post-infection or post-vaccination, are capable of neutralizing the new virus, new variant, to the same extent as the old variant. So that testing needs to be done, and not as one or two representative samples, but a respectable number. And that can be, kept available. So that's one point where virus numbers required will be somewhat large. The second point is again for transmissibility, which is sort of any cell line which is expressing ACE2 can be infected with the old virus and the new virus and newer virus to check how efficiently the infection is taking place. All of this will take, in my judgment, a minimum of 3 weeks to a month if we are ultra efficient and if there are no hiccups.
Vinod, you want to add to that?
Yeah, so just a quick point to add. So there are essentially two things that we're looking at. One is transmissibility, the other one is immune escape. Now for transmissibility, of course, you need to have experimental proof, but of course, there's surrogates to that, also mathematical surrogates to that. But in terms of immune escape, I think we are pretty much big fortune in the sense that a significant number of people are already vaccinated, at least 20 to 25 people. So one of the quick things to go back and look at is to look at the prevalence of breakthrough infections caused by a particular variant, which is emerging. So if you have a prior, for example, Delta, you do have a prior of what's the percentage of people who could have a breakthrough infection. So if you could look at the emerging variant and see if there is a more than expected chance of affecting breakthrough infections, then we can sort of suggest that this is possibly an immunostate mutation. So that's pretty much quicker because you just need to follow up the patient back and ask the vaccination dates.
Okay, with that, we come to the end of this program. Thank you all very much on my behalf for a great set of answers, a great set of presentations. Rahul, do you have anything to say? Do you want to wind up? Can't hear you.
Why is he following me when I was no need to do that.
I have no idea what's happened. So anyway, let me just...
So instead of Rahul, I'll just do the job. Okay. So thanks to all the speakers for promptly accepting our invitation for participating on this symposium. Thanks to Gautam for doing a wonderful job at moderating the discussion. Thanks to all of you for the wonderful presentations and the great discussions that we had just now. And thanks to all of you who silently listened to all the talks, provided questions, and for this nice discussion after the presentations. Thanks to our director for encouraging us to have this symposium. And thanks also to our outreach staff, especially Varuni and Gayatri, for organizing all the necessary tasks so that this event is successful. So thanks to all of you, and hopefully we'll have other meetings like this in future. Thank you.
Thank you.
Bye. Take
Thank
care
you.
and good
Bye.
night, everyone.
Bye.